Hello, AP Bio students, and welcome to an overview of Chapter 15, Gene Expression. I believe my slideshow should be starting. We need to understand the differences between prokaryotic and eukaryotic gene expression and how these two different types of cells and two different types of organisms can respond to changing environments and express proteins and traits at different levels and at different rates, and also at different stages within the transcription and translation stage. Generally, what we need to know is that there's going to be some level of feedback inhibition and also multiple genes within any type of metabolic pathway. So if we're looking at the synthesis of tryptophan, tryptophan might be made in cells when there are low levels of tryptophan from some precursor molecule that will be reacted by enzyme 1, which is coded for by TRPE and TRPD gene. And then enzyme 1 will turn that precursor molecule into this molecule, which will be reacted by enzyme 2 into this molecule, and then finally reacted again by a third enzyme, which creates tryptophan. And each of these enzymes that are proteins with specific structure and function, with active sites that can only react with molecule 3, 2, or 1, will be coded for by DNA that is translated and transcribed. But also, we're going to have some level of feedback inhibition. Thinking back to cell signaling, we know that enzymes can be allosterically regulated by molecules that bind either at the active site or not. It would make sense to not express genes if we have a high level of tryptophan because we don't need to do this metabolic pathway. Which brings us into the notion of operons. Operons are a group of functionally related genes that can be sort of coordinatedly controlled by a single on-off switch. Think about your kitchen and how you might have one switch for a bunch of different lights that turn on simultaneously. The regulatory switch is a segment of DNA called an operator, which is usually found within the promoter and tends to be near the top box in eukaryotes. An operon is the entire stretch of DNA that includes the operator, the promoter, the terminator, and all of the genes that control the functional products of our trait. Now, operons can be switched off by a protein that's called the repressor. And the repressor protein prevents gene transcription by binding to the operator and sort of creating an allosteric regulation of DNA that prevents RNA polymerase from binding. The repressor is going to be the product of a separate regulatory gene that might occur downstream, upstream, or on a totally separate chromosome that is also itself regulated. The repressors can be in an active or an inactive form depending on the presence of other molecules. We'll talk more about that in terms of the differences between repressible and inducible operons. A co-repressor is a molecule that cooperates with the repressor protein to switch an operon off. So for example, E. coli can synthesize the amino acid tryptophan in environments that are low in tryptophan. We will have our operator that is found within, Claire's saying, I hope I have a good morning. I'm going to turn that off. Do not disturb. We have an operator that is part of our operon with our 5 different functional genes that make polypeptide subunits that make up enzymes for tryptophan synthesis. We have our promoter that's going to bind to the beginning of the coding strand because this is a 3 to 5 prime. And up over here we have our regulatory gene that makes a 5 prime to 3 prime that makes a protein that can inactivate the repressor. So this is an inactive repressor that could become activated by binding to the operator and preventing the promoter region from binding. So when we have tryptophan present, tryptophan is going to bind to the repressor molecule, which will make it active, and then this activated repressor will prevent RNA polymerase, which attaches at the promoter, from interacting with the DNA molecule. A repressor operon is one that is usually on, and the binding of a repressor to the operator shuts off transcription. The TRP operon is a repressible operon, which means most of the time it is going to be turned on because E. coli are going to be found in an environment that is low in tryptophan, hence they are going to need to make more tryptophan. Alternatively, we can have inducible operons, which are usually off. A molecule called an inducer inactivates the repressor and turns on transcription. Lac operon is an inducible operon that we find within enzymes, or that is found within E. coli that contains genes that code for enzymes that are used in the hydrolysis and metabolism of lactose. So inducible operons tend to make things that break down molecules. By itself, the lac repressor is active and switches the lac operon off. A molecule called an inducer inactivates the repressor to turn the lac operon on. So in this case, we have our LAC-I regulatory gene that makes an active repressor that is going to almost always bind to the operator. And this is a inducible operon, so we have to turn it on. It is usually off. When the operator is connected to the repressor, RNA polymerase is not going to bind, so we make no RNA. However, in the presence of allolactose, which is an inducer, allolactose being an isomer of lactose, the promoter region or the repressor molecule binds to allolactose at its active site and it becomes inactive, which means RNA polymerase can make some different types of enzymes that are going to be responsible for breaking down lactose, so the metabolism can persist. If lactose metabolism persists, lactose being a disaccharide, and lactose levels fall, we would then expect allolactose to not be able to bind to the repressor molecule, which means that the active repressor would then prevent further RNA polymerase activity. So inducible enzymes usually function in catabolic pathways, meaning that they break things down. Their synthesis is induced by a chemical signal. When it occurs, it generally means that there's a high level of the molecule that we want to break down, such as lactose. Repressible enzymes usually function in an anabolic or build-up exergonic reaction. Their synthesis is repressed by a high level of the end product, meaning we don't need to make any more of it. Regulation of the TERP and LAC operons involve negative controls of genes because their operons are switched off by the active form of the repressor molecule. So again, this is going to be turned off with the active form of the repressor. And when we go back to E. coli, this will also be turned off when we have a co-repressor molecule bind to it. It's just that in this case, this is the end product for a catabolic pathway, and for an anabolic pathway, For a catabolic pathway, lactose is going to be the initial product. Now, E. coli will preferentially use glucose when it is present in the environment because it's simple, it's easy to digest, there's not a lot of enzymes active, it is the main source of ATP. But when glucose is scarce, CAP, a catabolite activator protein, acts as an activator of transcription. And CAP is activated by binding with cyclic AMP, which is a secondary messenger that we've seen before in cells. The activated CAP attaches to the promoter of LAC operon and increases the affinity of RNA polymerase, thus accelerating transcription. So if and when we have low levels of glucose, E. coli cells are going to do their very best to make as much lactase as possible to continue cellular respiration. So this inactive CAP molecule, which is signaled by increasing levels of CAMP when glucose is low, will bind to the promoter and also help with RNA polymerase. On top of that, we will have our repressor molecule become inactive with allolactose and bind to the operator to also, or it won't bind to the operator, which means that it won't prohibit RNA production of the LAC operon. But if we do have low levels of, or if we do have lactose present and glucose is present, that inactive CAP won't bind to it, so RNA polymerase will still likely bind to it, but it won't do as much production. So we can see sort of a gradient of lactose operon production based on the amount of glucose and also the amount of lactose. Now, eukaryotic gene expression is a bit more complicated because it's regulated at many stages. All organisms have to regulate gene expression at any given time, but in multicellular organisms, it's really key for cell specialization and tissue function to have lots of endocrinic signals going between different cells to regulate gene expression. This is a little bit messy because the wording on this PowerPoint is a little off, but generally we can have modifications of the chromatin, DNA, mRNA expression, and then also post-mRNA transcript modification. So there's a lot of ways that we can modify the gene expression in eukaryotic cells. We can then take that mRNA and we can degrade it after we make polypeptides. We can have those proteins be processed and cleaved and chemically modified in the Golgi apparatus, which makes our active protein. Sometimes we might not need those proteins, so we degrade them again. And then finally, proteins can do their final function, such as a phenotypic expression, enzymatic activity, structural support, becoming a signal molecule. So there's a lot of mechanisms in which we can regulate gene expression in eukaryotes, all the way from DNA to protein expression. One of the ways that we can begin to regulate DNA expression and gene expression is the change in histones. And histones are protein structures that build up the initial chromatin fibers in DNA. Histones being a protein structure that are positively charged, so they interact quite well with the negative charge of the phosphate backbone in DNA. In histone acetylation, we add acetyl groups to the positively charged histone tails. And this generally loosens chromatin structure, which promotes the initiation of transcription. Alternatively, we can add methyl groups to these lysine tails, which can condense chromatin and lead to reduced transcription. So when we acetylate histones, we're unwinding the chromatin fibers, which allows DNA to be more easily read by RNA polymerase. Secondly, we can have many different types of control elements. We can have proximal and distal control elements, which are upstream of the promoter, and these generally are where very specific transcription factors that are themselves proteins regulated by genes can bind to and either open up or close down DNA from being expressed. And we can also have the RNA processing that occurs where we take out the introns from one mRNA product to make a fully exon-based protein structure that is going to be made into a specific protein. And we obviously know that the 5' cap and the poly A tail prevent mRNA degradation in the cytoplasm. So activators can bind to the DNA strand, and the DNA strand then sort of bind based on DNA bending proteins binding to DNA, and a group of mediator proteins that bind to the enhancers. And this is sort of going to be the primer for general transcription factors to come in. And once the general transcription factors bind to the promoter region of the DNA section that we're going to code, then RNA polymerase II can bind in. General or mediator proteins are very specific for each gene, and they allow the general transcription factors to help further bind and open up the DNA to be transcribed into mRNA. And this is really important for eukaryotic cells where we have the same genetic information in, say, a lens cell or a liver cell, but we might not want to be producing or expressing the same genes. So the available activators in a nucleus of a liver cell will be much different than a lens cell, which means these proximal control elements will help the transcription factors bind to DNA. Well, alternatively, different control elements will bind to the regions of the DNA, which will allow crystalline gene to be expressed in the lens cell. So this is a really good way of either turning on or off different genes by having these enhancer control elements either be activated or not. Obviously, we know that we can splice RNA, so that means that we can make many different types of proteins from one RNA product, depending on whether or not we identify different segments of DNA as introns or exons. So we can make a 1, 2, 3, 5 or a 1, 2, 4, 5 protein by splicing different segments of RNA. And we do this by creating different types of spliceosomes in different cells by themselves being regulated by different genes. So all of these things come back to other genes being expressed simultaneously to produce these control elements, these transcription factors, or these spliceosomes that then go interact with very specific RNA products within very specific cells. The last thing we need to talk about is non-coding RNAs, and non-coding RNAs are important within the human genome. Only a small fraction of DNA in our body encodes for proteins, and only a very small fraction of non-coding proteins consists for genes for making things like rRNA or tRNA. So a significant amount of the genome is transcribed into what we call non-coding RNAs. And non-coding RNAs help regulate gene expression at several points. We can have microRNAs or miRNAs, which are small single-stranded RNA molecules that can sort of complementary bind to mRNA sequences. And when these things bind and form complementary pairs, it can either block or degrade the mRNA, preventing translation. And miRNAs are also expressed by gene regulation. So the mRNA is going to be produced, it's going to associate itself in a protein complex. We're going to have the complementary pairs form, and this is going to be very specific for each type of gene. We need to have a perfect match up to at least 9 nucleotides in a row for mRNA to be degraded. Hence, we are not going to allow it to be produced. Alternatively, we can just prevent the ribosome from seeing the mRNA product, so translation is blocked. We can also use mRNA products to understand the expression of different genes in different cells. So let's say we're trying to figure out what genes are activated in a liver cell or a skin cell. We can take mRNA products from the cytoplasm and do a product called reverse transcriptase. That is going to be where we take our mRNA product and we use a DNA, or we use an enzyme called reverse transcriptase, which makes it the non-coding complementary strand of DNA from our 5' to 3'. So it's going to be making our first 3' to 5' molecule, and then we can use DNA polymerase to make the 5' to 3' strand. That allows us to make cDNA, which has a complete coding sequence without any introns. And that's important because that allows us to see how also the same gene might make different proteins in different cells. We can take that cDNA and then we can apply it to a gel electrophoresis and compare different cells at different stages in an embryo or different cells within different individuals and see the results of how they match up between each other. So we're going to take the mRNA, make cDNA, and then probably look at different organisms or different cells or different parts of a body to compare relative expressions of genes. And we can do this also with radioactive dyes to see which genes are expressed in different tissues, where different colors mean different things in terms of gene expression. So this is called a DNA microarray. If we see red dots, it means that genes in the red wells are being expressed in the first tissue. And we're doing this by adding a fluorescent dye that matches with the RNA product. The same thing would be for green and yellow If we see black, it means the genes are not being expressed in either tissue. And we use this often to do biopsies of individuals with cancer to see which cancerous genes are being expressed or not in a whole host of different tissue samples.